Yes, yes. It is that time again. Time for another lecture. What you've all been waiting for. Okay, that's my high-tech trick for the day, I guess. So today, we're going to be talking about ancestry, ethnicity, and genetic testing. Mainly about ancestry and genetic testing. Um, this particular lecture is directed at all you curious folks who may ask the question, wait a minute, if race isn't a real thing biologically, then how come that one test that I took or that my cousin took said that we're 15% Italian, yada, yada, yada? So how can these two things both possibly be true? So that's what we're going to talk about. And it's very complicated. This is largely based on what I know about what Ancestry DNA, that company, does in their ancestry testing. So they put out a white paper, which is a technical explanation of their methods. And so this is largely based on that. Other companies will have different methods, but they follow similar principles. Okay, so I might have to break this into a couple lectures. We'll see. First of all, people get the impression that, sorry, I realize I move around a lot in these videos. People get the impression sometimes, let me go back here, that, what you are, what your biological makeup is like these cans of paint. Like, oh, I've got some of that orange and some of that yellow and 20% of that aquamarine or whatever. But it's actually quite a bit messier. It's quite a bit harder to pick apart. And actually, that palette there doesn't even really do it justice. It's really like this muddled mix of stuff. Because there is no color. There is no, in this metaphor, there's no, like, Irish gene. There's not that one gene they're going to find and it's going to tell you unequivocally, oh my God, you could have only gotten this from the leprechauns or something. It just doesn't work like that. So it's all about percentages and statistics and boring stuff. And it's really complicated. Yeah, I forgot how many of these pictures I actually put up here. So before I get into it, let's just go over some things we already know. One, our species evolved in and spread out both within and outside of Africa. So it all started in Africa for all of us. People move and mix around and get it on all over the world and kind of always have been that way. It's really just a matter of how much more we're moving around now. But people, even if they didn't move too far from home before, there have been lots of periods of migration and mixing and gene flow. Okay. So we talked last time about population structure, and population structure is just how within any population, however you define that, even if you're going to stick to the island of Ireland or some small population, you're going to find structure where there are certain groups that may share slightly more genetic affinity, certain areas where it might not clump together quite as much. So there is that sort of thing, and there's also that sort of thing globally, but there's not population structure to the degree where there are these separated, isolated groups that we would call races, or even groups that are obviously, different enough from humanity as a whole to be called races. So that's a level of structure that we just do not have. Our species is far too young, and we mixed and moved far too much for that to have occurred. We also know that genetic distance, which just means how genetically different any two populations are, is proportional to their geographic distance. Okay, for an explanation of that, see the previous couple of videos. It should be fairly obvious that throughout most of human history, the people that you were most likely to have children with were those who lived pretty close to you. Now, there's still sort of a domino effect, sort of a chain reaction where genes can spread over large distances, even if people are only spreading over small distances, because you have children with someone in the next village, maybe your children grow up in that village, they have children with someone in even the next village over, and so on and so forth. So genes sort of get carried all around the world, faster or slower, depending on how much people are moving. And as I said already, there is no Irish gene, there's no Nigerian gene, there's no Thai gene, there's no identity gene. And this is not going to give you your identity. Identity is a cultural thing that we sort of create as a society and as individuals. So while Ancestry can tell you where your more sort of recent ancestors came from in history, it can't really tell you how to identify or even how they identified. So there are some assumptions behind the testing. One assumption is that if you take any particular section of DNA from two different people, it's probably going to be, those sections are going to be more similar if those people come from a region that's pretty small or pretty close to one another than it is when you're comparing two people from far-flung regions. Okay, that goes along with what we know about genetic distance correlating with geographic distance. Okay. Also, another assumption is if we have enough data from people from many different populations, we can probabilistically determine the likelihood that a particular stretch of DNA with a particular sequence comes from an ancestor who lived in a particular region. Okay. So while there is no barcode that will say, that's the Irish gene, if you look at enough people in Ireland and enough people in England and enough people in France or whatever, and you compare them, you can start to see some statistics that say this particular combination of alleles is most likely to come from someone in the Irish population. When you're getting to populations that are super close together, both geographically and in historically, in terms of migration and mixing, like Ireland and England, for example, you have to have a lot of data before you can pull those apart. Yeah, it's like trying to tell identical twins apart. Their parents can do it. They can do it. Obviously, hopefully, close friends can do it. But the more similar two people are, the more kind of points of reference you're going to have to be able to kind of pull them apart. Now, it should be noted that at any particular locus, which means one location in the DNA, it's totally possible that two people from way far-flung different regions have the same allele. All right, so an allele that, especially an allele that's neutral, is selectively neutral, that doesn't have an impact on fitness in a particular environment. It may be found at highest frequency in West Africa, but you can probably still find it in Japan. Okay, so things are not as separated as we may assume. All right, so the first thing they have to do is they have to have a reference sample so that they can determine the frequencies of different alleles in different populations. Okay, so what they do is they get a reference sample of contemporary people living today in these particular populations, however you want to define a population. Okay, could be Ireland, could be the British Isles. I know Irish people hate me for saying that. Could be France, could be Western Europe. You could define it smaller or larger, however you want. But you need a sample of people living in that population today. And you want to make sure that your sample doesn't include folks who recently migrated to that region, because when we're estimating ancestry, we want to try and get some time depth. We want to try to go back in time a little bit, right? So what they try to do when they get these reference samples is they ask people questions, and they ask, like, you know, where were you born? Where were your parents born? Where were your grandparents born? And they only generally they only accept samples from people who can trace back at least to their grandparents in a very particular area, okay? So the sources that they use come from a couple of open source studies, the Human Genome Diversity Project and the 1,000 Genomes Project. And those sort of paradoxically, the 1,000 Genomes Project has 1,800 samples, whereas the Human Genome Project has 1,000. And they come from a number of different populations around the world. And then Ancestry DNA, a particular company, has its own proprietary reference sample, culled, I suppose, in part from customers, and maybe they went out and did some of their own research as well. But each of these companies is going to like have their own little kind of secrets that they don't share with the other companies, right? And as I said, Ancestry DNA customers, I don't know if that's different from the reference sample, if it's part of the proprietary reference sample, what have you. So, like I said, these individuals can trace at least their grandparents to this particular region. And these are all living individuals, right? So it's a flawed kind of assumption. I mean, for the level of analysis that they're doing, it's fine. Because, you know, people didn't move around as much even a few generations ago as they do now. But they're not using ancient DNA, right? So if it says like, oh, you're 10% Egyptian, that doesn't necessarily mean like, oh my God, like that means the pharaoh in whatever period was my great-great-great-great-great-great-great-great-great-grandpa. Although probably he was, because everybody is related, especially, never mind, that's for a different lecture maybe. But anyway, okay, it's living individuals. Now, they have to also do some quality control on this reference sample. So they call 300,000 SNPs throughout the genome. Let me break that down. You may remember that a SNP is a single nucleotide polymorphism. So it's a one letter change in the genetic code, right? And calling a SNP just means the software and the machinery that they have chemically sort of analyzes this, and based on a signature that shows up, like a chemical signature, it says this is most likely a C, or this is most likely a T, right? If there are individuals who have missing data, not because they're missing letters in their genetic code, but because, you know, if you've ever tried to run like an actual experiment in like a chemistry lab or a physics lab or whatever, it never goes 100% as planned, right? So it may be that, I don't know, they had some hamburger or something for lunch before they spit in a tube, I don't know, and it messed things up, or the sample got degraded, you know, before it got to the lab and got tested. If for whatever reason it was difficult to call one of the SNPs, you know, that can result in some missing data. So they get rid of individuals that have too much of that. And they look to see if there are any samples that look like, whoa, just like complete outliers. So, you know, maybe you thought that your grandfather lived in the same village in Tuscany, but really, that wasn't your grandpa. You know, maybe your grandpa came from somewhere else. You know, maybe your grandpa was from, I don't know, India, and you know, the family just kind of kept it secret that there was a little indiscretion there. For whatever reason, they sort of remove these outliers just in case they're not representative of the population. So they do something called PCA, Principal Components Analysis, which is a statistical technique that takes all of the variation, you know, looking at all these different genomes, and it without explaining like all the ins and outs of it, it basically looks at kind of the viewpoint that shows the greatest variation in that and projects it two-dimensionally. So this is an example of PCA here done from Europe, right? And they, so this right here is supposed to represent like an Italian population. Well, they might look at, this little square over here and be like, I don't know, that one seems a little bit far removed. Might not actually be from an Italian individual, whatever that means, someone who's lived there for a long time, and they get rid of it. They divide their samples into populations based on clusters. So let me go back here for a second. So they might look at this data and they color code it based on where these people say they and their families are from, right? And some of them are pretty well-defined clusters. Like here, I suppose this looks like it's Finland. Okay, so boom, Finnish population. It's distinctive enough from the rest of Europe that it kind of clusters nicely. And so we feel confident that if somebody has a good amount of Finnish ancestry, we could correctly assign that to them. So that's a cluster. Now, let's see. England, Wales, where's Ireland, Scotland? Like, so England, Wales, Ireland, Scotland, like that's all like around here. That's all kind of in this big soup of stuff that you can see it doesn't cluster very nicely. There's a lot of overlap between England and Wales and Ireland and Scotland. And so they may say, we don't really have enough confidence to separate those out into two distinct groups, so they might just call them British Isles, right? In the case of Ancestry DNA, they feel confident enough, so they lump Ireland and Scotland together just because they're much more difficult to pull apart because of a shared history. And they lump England and Wales together as well, okay? So they divide these samples into populations based on clusters. Then within those clusters, they define haplotypes. And a haplotype is a little hard to explain, but let's take a stretch of DNA and let's say there are 4 SNPs, that happen in that stretch of DNA, and for each of those locations, those 4 locations, there could be maybe 2 or 3 different letters that could be there. So a haplotype is defined by a stretch of DNA with a certain number of SNPs, right? And they'll say, if you have an A, a C, a G, and a T, that's one haplotype. If you have an A, a T, a C, and a T, that's another haplotype, right? Now, there could be other variations in there because mutations occur fairly regularly, but just to kind of look at who inherited this same stretch of DNA, they define it as a haplotype. And they calculate the frequency of that haplotype and of all the ones they defined within a population, okay? So haplotype 1A, frequency in Ireland, 15%. Frequency in England, 20%. Haplotype 1B, frequency in Ireland, 10%. Frequency in England, 13%. So that's going to allow them to do some statistics to determine which population you likely inherited a stretch of DNA from. If you want to watch this video, it'll be on the PDF. Okay, it's a good short video about haplotypes. And then they go back and they test their ability to correctly predict the ancestry of individuals from the reference sample. So what they do is they take 5% of their reference sample and they run it through their algorithm that they've developed based on the frequencies of these haplotypes. And they let the algorithm sort of look at all the haplotypes and look at the frequencies in different populations and come up with a mixture of ancestral populations for that individual. Now, I guess for these individuals in the reference sample, they're supposed to really only have one ancestral population. So in this case, they're trying to see, can they correctly identify the population that individual came from, right? They do it 20 times, and they actually give you a graph of how accurate they are for different populations. Pardon me while I have a sip of water. Refreshing. Now you notice there's some differences here. So for North Africa, for example, in this box plot, this is the average. So out of those 20 trials, on average, they correctly estimated about 60% of that person's ancestry as being from North Africa. Not great, not terrible. Now, down in Nigeria, if you look at this box plot for Nigeria, they, on average, a person from Nigeria in their reference sample is only going to, show up around 30% of their ancestry from Nigeria, all the way up to like 60%. Okay, so that's not fantastic. Now, some other populations, like, let's see, Ashkenazi Jewish or European Jewish, they have a pretty high success rate of putting someone correctly in that population, as they do with Finland. But then, there's some other ones like, let me see, Spain, not super great down here. And Basque population, strangely, also not super great. So they just don't have enough data from those regions, and or they are too similar to neighboring regions to really pull them apart with a great degree of accuracy. So like I said before, if a population, if the estimation doesn't do well for a specific population, they might pool it with the neighboring populations, right? So in that scenario I showed you, they could lump Spanish and Basque individuals together. I believe that they don't do that, but they could do that. And it would be more accurate to say, well, you're either Spanish or Basque or a little bit of both. So like I said, they have different amounts of samples from different populations, and that's for a variety of reasons. One of those reasons is that, well, at first anyway, the majority of people buying these ancestry tests were white people from the United States, right? So from an economic standpoint, that was their market, so they got a lot of data from Europe. The other thing is a lot of the scientists that had started doing this kind of research were European or white American, and sort of probably defaulted to that bias. But also, it's easier to get samples from certain places than others. For example, you might understand why some Native American communities might be hesitant about giving their DNA to a scientist, especially if the scientist happens to be a white scientist, just because of the history, and because of legal issues around who can belong to, who can be considered part of what tribe. So there's socio-political things behind this as well. But basically, in their current reference sample, they have almost 17,000 total samples. Now, this is based on a white paper that came out a few years ago, so it might be different now. It probably is a little bit higher now. They have defined from those samples 43 overlapping regions. So, they're not completely bounded. They overlap to some degree. And here you can see the number of samples for each cluster or population as they defined it. So Northern Africa, they only have 41 samples. That's why they're not doing Algeria, Morocco, Tunisia, Libya, right? How can you divide up 41 samples into a number of different countries and expect that that's going to be representative of those countries? You just can't. Africa, South Central hunter-gatherers, 34. Wow. Cameroon, Congo, and Southern Bantu peoples. That's a lot of people. But they have 579, but they're representing a lot of different sort of identities in a broad geographic region. Now, if we look at England, Wales, and Northwestern Europe, 1,519. See, that's a lot of samples. France, 1,407. Germanic Europe. What was that? Germany, Austria, Liechtenstein. I don't know. Are they Holland, maybe? I don't know. But anyway, they have 2,072 samples from there. Okay. So you can kind of go through this and see for yourself what the makeup of their reference populations are. You know what? This might be a good time for me to cut this off because I'm not sure how long I've been talking, and I will be back with the second version, second part. Okay? See you soon.